I would like to welcome our first speaker, who is our keynote speaker, and this is Dr. John Spiro. He's giving a talk today on how fast laser focus benefits translational efforts in neurodevelopmental disorders more broadly, the view from a large private research funder. And so what I want to share with you about John is the Senior Vice President and Senior Scientist at the Simons Foundation, the Simons Foundation Autism Research Initiative, or SAFARI. And that may not mean a lot to you guys because that's a lot of words, but what I want to share with you is that from a scientist's perspective, so the mission of the Simons Foundation, which is a nonprofit foundation that their mission is to advance the frontiers of research in mathematics and basic sciences. And really, they're a nonprofit champion of basic science research. And as a scientist, it's equivalent or maybe a little bit more prestigious if you get a grant from the Simons Foundation as if you get a grant from the NIH. And so people work really hard, and when they get grants from Simons, they're like on another level because they are so critical and rigorous in the science that they produce and what they fund. And I remember when I was invited by the Azoots to come to the Simons Foundation with Roy and meet Jim Simons, who's the founder, and his wife Marilyn, and John. And I walked in the room, and I didn't know any better, and Roy is like, these guys are like a big deal. So, we have to be prepared. What are we going to say? We have to make sure they understand Angelman syndrome. And I was all nervous, and I was all ready, and I was prepared. And I met John, and he just calmed all of my nerves. And I was under the very strong impression that, we can't say the word cure. We can't think about brain research and neuroscience in a way that we're going to actually necessarily transform it on the basic science level. We have to understand more. We have to do more research. And after talking to John, and our relationship has blossomed since then, which he'll talk about from that first day that I met him, what I realized was that we are so aligned from a basic science perspective to a translational research perspective once we can meet in the middle ground and realize the value of the entire continuum of scientific research. And they are incredible scientists that they fund and that they work with in their own institution. And for him to come here as our keynote and be able to say that I am proud of what FAST and the Angelman syndrome community is doing is such a humbling honor. So John, I'd like you to come take the stage.
Thank you, Allison. Yeah, I'm mic'd up.
How to use that.
Yeah. Hi, everybody. It's an incredible pleasure to be here, and it's a real honor to be introduced by Allison in that way, so it's mutual. As usual, it probably would be better for me to just step back and have Allison give my talk. Let's see if I get this right. There we go, whoops. So the textbook story of how science works often gives the impression that science is sort of a slow and steady accumulation of interesting facts. But I think when you look more deeply at how science works, it often is the case that there's sort of tipping points. There are key experiments or clinical trials or key insights that not only change the way that people think about the field, but bring all sorts of new money and new talent into the field that really rapidly accelerates the progress. And so what I thought I would do here is tell 4 short stories of tipping points in the history of neurodevelopmental disorders. I'm going to start somewhat immodestly by talking about the Simons Simplex collection, which was the first big project at the Simons Foundation. Then I'm going to talk about a so-called reversal experiment in a mouse model of Rett syndrome. I'm going to talk about ASO gene therapies for spinal muscular atrophy. And now, major spoiler alert, lastly, I'm going to talk about how what I think is happening right now with the Angelman community and FAST in particular is the emergence of another one of these tipping points. So let me start by giving you just a few words of my background. Allison already said some things. First of all, I guess I'm supposed to say I have no conflicts to disclose. So I was trained as a very basic neuroscientist. I got a PhD and a postdoc studying very basic questions about how neurocells work and how they form circuits leading to behavior. And instead of pursuing an academic track, I became sort of a professional generalist. I worked for several years at Nature Neuroscience, which at the time was a new journal. And then I went and became the neuroscience editor of Nature. I'm just giving you a little context of where I come from. And then I had the really good fortune to join the Simons Foundation in 2007. As Allison said, this is a foundation. It's a private foundation based in New York, founded by Jim and Marilyn Simons. Jim is an incredibly distinguished mathematician turned hedge fund investor and made an enormous amount of money that he's been giving away through the foundation, which has a very simple mission to advance the research in basic science and mathematics. But as Allison mentioned, there's a division of the foundation called SAFARI, or the Autism Research Initiative. The mission is to improve the understanding, diagnosis, and treatment of individuals on the autism spectrum. It was launched in 2003, and we're incredibly privileged to have a very generous budget in the range of about $110 million this year. We don't have to fundraise. We don't have to sort of, it's an incredibly privileged position to be able to take this long view on how to help the field. Just stepping back one slide just to give you a sense of the foundation more broadly. When I joined, I think I was employee number 6 or 7. There are about 500 people that work there now. And it's sort of divided into 2 halves. There's a grant-making half, and SAFARI is part of that. And then there's an internal sort of intramural computational unit called the Flatiron Institute. So it's become a really interesting place to work. So let me start this story by telling you a little bit about the Simons Simplex collection. So let's back up and talk a little bit about how autism spectrum disorders are diagnosed. It's important to know that they're really diagnosed by behaviors. It's a book called the DSM-5. It gets updated every couple of years. And in order to be diagnosed with autism, you need to meet criteria that are listed below, or at least some of these criteria, such as persistent deficits in social communication and social interaction, restricted repetitive behaviors, et cetera, et cetera. But when we started around 2007, we knew that autism had a strong genetic component, and we knew this based on some really beautiful twin studies where they looked at the rates of autism. If one identical twin has autism, what's the chance that the other one does? And it's very high, in the range of 70 to 80, 90%. It's the most heritable neuropsychiatric disorder in that way. Fraternal twins are lower, excuse me, and siblings are lower, but still quite elevated. So we knew that genetics were involved, and we knew that we knew one or 2 genes like Fragile X or MECP2, the gene that underlies Rett syndrome, was involved. But we really didn't know anything else about the genetics. And at that time, Simons decided to take a really big bet on genetics. And this is one of the things we can do at a private foundation, where we can just decide essentially to really double down on something. So we launched something called the Simons Simplex Collection. And this was an open science project. I want to stress that. All the data and everything were freely available to the whole research community. It had a simplex design, and let me just explain that. So we focused on families. There had been a long history of discovery of autism genetics that frankly was not very productive. So we took a bet on focusing on a simplex design. So that's a family that has one kid with autism, but everyone else in the family was neurotypical, including another neurotypical sibling, and that ended up being a really important design of the study. We spent a lot of time and a lot of money standardizing data and collection and specimens, and then we got extremely lucky because along the way of putting together this collection, the price of genomic analysis fell precipitously. So when we first started, we had ideas on how we'd look for genes, but when the price of whole exome sequencing and other types of technologies came on board, it allowed us to do it fully unbiased. So instead of starting thinking where we might look, we looked across the whole genome. And now I'm going to fast forward about 15 years, and of course I don't expect you to read this very busy slide, but the takeaway is that we found an enormous number of genes, really quite surprisingly. And I just want to be clear, some of these genes, like Fragile X or MECP2, some of these genes increase the chance of having autism or related neurodevelopmental disorder by like 100 or 200 times. These are not small effect genes. We also found a large number of so-called copy number variation regions. These are regions of the genomes where small pieces of DNA are either duplicated or deleted. So this, I think, really became a tipping point in neurodevelopmental disorders. First of all, this discovery of all these genes, and I should be clear, the Simon Simplex collection started this off, but a lot of other people moved into this field after we started it. So it brought an enormous amount of new talent and money into the field of neurodevelopmental disorders, and I think this was key to really turbocharging things. It was clear that understanding the genomic architecture by knowing these genes was going to lead to a rigorous way to understand autism biology and heterogeneity. So the genes would lead us to the really interesting biology, and this is still very much an emerging story. It's really important to note one of the key discoveries, I think, which we're still coming to terms with, is although we were looking for genes in an autism cohort, it turns out that many of these genes that we found are also risk factors for other disorders, such as epilepsy and intellectual disability. And I think this is where the biology is teaching us that that DSM book that I showed at the beginning was not written by some superpower. This was a human attempt, and we have to kind of keep thinking about this. So it raised many new questions. What are these proteins that these genes do? What are the proteins that these genes encode do? Where are they active? Who are their interaction partners? I want to just be clear before I move on to the next story that the genetic landscape of autism is actually even more complicated than I've let you know. There are a whole bunch of polygenic risk factors and other things, but these highly penetrant genes that I showed you are really key to understanding the biology. So I want to move on now. Once we sort of started to understand the genetic landscape, I want to move on to an experiment that's commonly referred to as a reversal experiment in the Rett mouse model. So I'm showing you a paper here from Adrian Bird's lab in Edinburgh, and incidentally, without my knowledge, I realized that one of the 4 authors of this paper is sitting right over there, Stuart Cobb. I saw him yesterday and realized that. So this is a mouse model of Rett syndrome, and it was made by deleting the MECP2 gene, which is the gene that, when mutated, causes Rett syndrome. And these mice recapitulate some of the human troubles that people with Rett syndrome have. They have motor and breathing issues and cognitive deficits. But the researchers, instead of just deleting the gene, did a really clever thing where they put it under a switch so that it could be turned on or off depending on what they fed the mouse. And they found something really remarkable. So normally, when the gene is deleted, there's symptom onset after several weeks, and then the mice get quite sick, and in fact, they die and have all sorts of other problems. But remarkably, if even after the animals are showing severe deficits, if you turn this switch back on, you can rescue much of the behavioral deficits. And the one slide I'm showing you there, the red arrow indicates when the gene was turned back on. And right underneath on the bottom right panel, it shows you essentially the survival curves of the mice. So the y-axis there is the % survival. And so basically, by turning this gene on, even at a late stage, they could see all sorts of changes in behavior and cognition that I'm not showing you, but this experiment is pretty striking. So I think this represents another tipping point in the history of neurodevelopmental disorders, because it told us that a supposedly neurodevelopmental gene has ongoing acute effects after birth, postnatally. And it was really, I think, an unexpected late rescue of some components that was possible. Obviously, this was in a mouse model. That was a really stunning result, and I think a lot of people in the field were forced to rethink the concept of a neurodevelopmental disease process. If it's a neurodevelopmental disease process, you wouldn't think you would be able to fix it at a relatively late stage. And so this raised an enormous amount of questions and generated a lot of enthusiasm in the field. would this be the case for the other genes? I just showed you on one of those previous slides a long list of genes involved in neurodevelopmental disorders. Would you be able to do the same thing in these genes in animal models? And could this be the case for humans? And so now I'm going to move on to the next story, which is using antisense oligos and then following that gene therapy in spinal muscular atrophy. So spinal muscular atrophy is a progressive monogenic motor neuron disease. The onset is pretty early during infancy, and it's pretty bad. So kids fail to achieve motor milestones. They might die or actually need a ventilator within a few years. And based on an enormous amount of preclinical work, investigators were able to design antisense oligos. You're going to hear a lot more about that later today. And then eventually a gene therapy that essentially replaced the broken gene, the spinal muscular atrophy gene, and this was in humans. So I'm going to show you just one slide that I think is really striking, that's burned into the memories of a lot of people who work in this field. This is from a paper that was published in the New England Journal of Medicine, and this is actually a gene therapy approach, but all the work that sort of built this gene therapy approach was done with antisense oligos. So what you're seeing here on the y-axis is a score that gives a sense of the motor ability of people. And then on the x-axis is the age. And what you see where the red arrow is when the kids were given the cDNA or the gene therapy. And what you see is that there's this very sharp increase in ability, largely motor ability. The dotted line around 40 is the level based on natural history data at which kids with SMA without an intervention would never surpass. So you can see that almost all of these 15 individuals got considerably better and passed that historical average. A lot of things have been written about this as a beautiful story in science. So I think this also represents another tipping point in the history of neurodevelopmental disorders. It introduced to the world a completely new class of drugs called antisense oligos, which again, you're going to hear a lot about today and more tomorrow. And the key thing is that they're targeted to the root cause of a genetic disease, as opposed to trying to target downstream effects of a genetic change, which are often really complex signaling cascades that might take decades or more for scientists to understand. So the key thing is you're going right after the genetic cause of a disorder, and you're not trying to chase the tail of all of these things downstream. So the ASO-based therapy, which was nusinerem or Spinraza, was approved in 2016, and I think it really set the stage for this gene therapy replacement strategy that I showed you on the last slide. And I think that's going to be a little bit of a theme in the field. So this raised some really interesting questions. Could ASOs and gene therapy be designed for other genetic changes? And would this approach work in the central nervous system? So spinal muscular atrophy involves nerves, of course, but it's really a neuromuscular disorder primarily. And it really was completely unknown whether you could get these drugs into the brain and have effects on really true central nervous system problems. The spinal muscular atrophy work also had another really interesting major effect on the field, because it was clear that earlier is better. So if you could identify kids pre-symptomatically before they started having trouble with spinal muscular atrophy, you could really help them much more than if you started helping them after the disease had progressed. And this got a lot of people thinking in really clever ways about whether genetic tests could be added to the standard newborn screening panels that are available in the United States in all 50 states, and these are available around the world in different cases. We organized a meeting at Simons about 2 years ago. My colleague Wendy Chung, who has since moved to Boston Children's Hospital, is leading some of these efforts. There are many, but this is an attempt to add genomic screening, such as whole genome sequencing or other genomic techniques, to the genomic panel so that we'll be able to identify kids earlier. There's a nice review that just came out talking about this as well. Okay, so this gets us to the spoiler alert story of the Angelman community and FAST in 2023. So I think of all the conditions, all of the genes that we've been thinking about, and geneticists are thinking how you might be able to target these genes, Angelman is really ahead of the pack in a pretty big way, actually. And there are a couple reasons. One has to do with the genomic architecture. And I noticed there's some beautiful pamphlets on the tables that explain this really nicely. But as many people in the room probably know, the paternal copy, the copy from dad of the UBE3A gene is repressed. So there's a strategy that almost reveals itself that if one could suppress the repressor or remove the brake and re-express that paternal copy, that one might be able to restore the UBE3A protein and essentially fix the problem. This is a really big advantage. Many of the other genes that I showed you are missing in ways that are more complicated to think of how you're going to fix the problem, but I think you're going to hear several talks today about other clever approaches which might be used in Angelman as well. But almost as important as the genomic architecture, I think one of the reasons why FAST is so, and Angelman is so far out ahead, is the group has just maintained this laser focus on setting the stage for translational efforts. And I can't emphasize how important this is. So the building a robust ecosystem of engaged families is clearly like a #1 priority. This is like, I'm just looking out here at everyone, this is clearly a critical component. Having natural history data and biomarker and outcome measure, the Biomarker and Outcome Consortium, so-called ABOM, is an enormously important recipe for setting the stage for success in this field. And just as one example, the development of the ORCA, this outcome measure for language, is exactly the kind of thing that the field needs to make progress. And then there's an enormous amount of preclinical work, and by preclinical, I mean work in mice, work in human cells, and things like that. But it's really, this is a moment. And so the tipping point here is that I think early evidence suggests that ASOs might change the course of a complex central nervous system condition by targeting the genetic cause. I can't emphasize how big a deal this is, and I think over the next 2 days you're going to learn about some of the really exciting progress on that. And this really opens an enormous question that's unknown to the field, which is, are cognitive language and motor changes still possible later in development? And I think, and this one is maybe more personal for me, but I really think this is going to start changing the field's thinking about how most effectively to use animal models. So I think it's actually creating almost a new playbook for how the field can proceed. And what I mean by that is we've supported, Simons has supported, I know FAST has supported a lot of work in preclinical models. It's absolutely critical to understanding the biology and thinking about rational therapeutic interventions. But I think what we're learning is that humans may actually be the best model. And when we have really safe drugs, or drugs that we think are really safe, like ASOs, it's going to be absolutely critical to do the proper experiments to get those into people and to learn from those changes. So I think we're going to see that small studies in humans are going to reveal critical issues not encountered in the preclinical work. So I want to be careful. I'm not throwing animal models and things like that under the bus. Those are always going to play a key role. But I think one of the key take-homes, and this has been a real learning experience for me, is how important it is to decide when to get into a human and when to start trying this. So I really applaud the community for this. So the rest of the neurodevelopmental field is watching really closely. Allison mentioned briefly that she and Jennifer and I have organized this meeting, I think we did it 3 years in a row now, called In Sync. And this is a meeting where we start first with representatives of the Angelman community, giving an update on the state of the science, not unlike what's happening here over the next 2 days, but in a sort of fast-forward version. And then we bring in other groups, other genetic groups that are trying similar things. They're trying to move from animal models into drug studies in humans, but we bring them in and try to help them benefit from the learnings of the Angelman syndrome community, and also figure out what they might have learned that the Angelman community might be missing, which hasn't been much, I would say, but it's been an incredible pleasure to organize this meeting. So I just have this slide here. I mean, I think it's really a case where Angelman is out ahead of the pack and is the position to be helping other groups sort of get up to speed. I just want to end on one word of perspective. So biology is really, really complicated. There's an enormous amount about the genome, and including, of course, the central nervous system, that we still don't understand in a significant way. One of my colleagues had this slide that I'm borrowing, where what we're talking about here, like the gene UBE3A, is really kind of the tip of the iceberg in terms of thinking how the genome is organized. So we're sort of going after that part of the genome, but just a reminder that there's an enormous amount of complexity about how the DNA is packaged and how genes get expressed. So just a little bit of perspective that it's complicated, and while we're making enormous strides here, that we have to sort of keep in mind this perspective. So I'm going to stop there. I really want to thank you for your attention. It's a great honor to be here, and thank you very much.